Hello, and welcome to your Chapter 13 lecture video on the cardiovascular system. In this lecture, we're going to cover heart and blood vessels, as well as concepts around blood pressure and cardiac output, those things. So just to start, to kind of give a brief introduction, and these first few slides I'm going to kind of go through quickly because we did cover a lot of this in lab. I'm going to go through this. You should definitely read through it, but I'm not going to linger too long. So the cardiovascular system is your heart and the blood vessels, all three types: arteries, capillaries, and veins, along with their subcategories. It's necessary for supplying oxygen and nutrients to tissues, removing waste from them, things like carbon dioxide, uric or nitrogen compounds, things like that, as well as like hormones and those kind of things. Arteries transport blood away, so away. Veins transport blood back to the heart. And capillaries are the ones that are in between. They help to, this is where actual diffusion of nutrients to the tissues and waste into the blood takes place, is at the capillary vessels. Two different circuits of the cardiovascular system. The pulmonary, that's where it's between the heart and the lungs. So where deoxygenated blood leaves the heart to the lungs, gas exchange occurs so that carbon dioxide can be exhaled out, oxygen can be diffused into the blood, and then it comes back into the heart to be pumped to the systemic circuit, which is the body, between the heart and the body, where you have oxygenated blood traveling along arteries to the tissues, and then carbon dioxide coming back from the tissues to the heart, through the pulmonary circuit, out through the body. Here's just a graphic representation of that. You should know all these steps, all these flows, everything that blood has to pass through. So the heart is a hollow cone-shaped cavity. It's right in the middle of the chest and what we call the mediastinum. That's the cavity, or the cavity in which your heart is resting. It's average size, about the size of a fist. It has a base that is the back of it, so that's on the posterior side, and then the apex is the point that kind of sticks out into the left lung. There you can see that. And here's just some other images showing you the pericardium around the heart, so the layers around the heart. So this is the pericardial cavity that it sits in. So the pericardium, peri around cardio, cardium heart, is a sac that holds the heart. It has two different portions. It has both the fibrous and the serous. So fibrous is referring to the connective tissue, whereas serous is a membrane, a serous membrane, like a mucous membrane. So this is the actual kind of layers directly around the heart. The serous pericardium has three features. Two of those are actual layers: the parietal pericardium and the visceral pericardium, also called the epicardium, right on top of the heart. And in between those is this potential space. It's called the pericardial cavity. So you have the parietal pericardium. If you remember, we did this. I know we learned this for the lungs. We talked about the parietal pleura and the visceral pleura. So parietal pericardium, pericardium around the heart. Parietal, it's telling you that it's going to be forming kind of the wall of the heart cavity. So it's more external. It's going to be brushing up against the fibrous pericardium. So the parietal pericardium is going to be brushing up against the fibrous pericardium. And then your visceral pericardium, the epicardium, this is the one that's directly attached to the heart itself. So visceral for your organ. And then again, in between those two is the pericardial cavity that has serous fluid that helps to reduce friction between those layers as the heart pumps, contracts, and expands. This pericardial cavity with its serous fluid helps to make sure that that action can happen smoothly. So both of these two pericardiums are the serous pericardium. So remember, serous is just a general name for a membrane. You can have the pleura in your lungs is a serous membrane. This one is a serous membrane. Serous, the difference between serous and mucus, if you forgot, mucus are external, so oral cavity, nasal cavity, reproductive, urinary tract, sorry, and then serous are internal. So looking at the wall of the heart itself, so kind of we started, larger, talking about the mediastinum cavity in between the lungs, and then now we're getting into the pericardial cavity, the pericardium, the actual sac around the heart, and now we're talking about the walls of the heart. So the epicardium, the visceral pericardium, that was again, part of the pericardial sac. So this is the layer that's directly on the outside of the heart wall. So it is both, this layer that we're talking about here is both part of the serous pericardium and the external layer of the heart wall. It's that outermost layer, that serous membrane. The one in between is the myocardium. So this is the muscle layer. It's going to be the thickest layer of the heart wall, particularly around that the ventricles, the lower half, and specifically on the left side, the left ventricle. This is really what's responsible for pumping blood out of the heart chambers. And then the endocardium is the inner heart layer. It's again another connective tissue and epithelial layer, so it kind of lines the inside of the heart as well as the major vessels where they join the heart. So it kind of creates this nice smooth passageway for blood to flow and slip through. The endocardium, so endo inside, the endocardium also has what's called the Purkinje fibers, and we'll learn about the cardiac conduction system here later in this lecture. But their Purkinje fibers are just a network of neurons that extend into the heart muscle to allow it to contract and relax at the right moments and pacing. Here's a depiction of the heart wall. You can see they kind of took this section. There's that fibrous pericardium, nice and thick around it. And then as you start to go deeper, you have the parietal pericardium, the pericardial space, the pericardial cavity, and then you get into the three heart wall layers itself. The epicardium, that thin pink layer, the myocardium, and then the endocardium, this other thin pink layer. So looking at structure of the heart has two chambers. The upper chambers are atria, the lower chambers are the ventricles. The atria receive, the ventricles pump. So the atria are receiving blood, returning from the heart, returning to the heart from the body or from the lungs, and they tend to have very thin heart walls. They're really not. They're only having to pump blood a little bit through the heart. So really, the atria are very thin walled, whereas the ventricles have that thick muscled myocardium. Separating them is a septum, so it separates the atria from the ventricle. I'm sorry, the two halves of the atrium. So the left and the right sides of the heart are separated by the septum. You can think, if you have seen me, I have my septum piercing, so it's that thing in the middle. So it's not unique to your nose or unique to your heart. It's just a description of a structure. Each side has valves controlling the flow of blood between the atria and the ventricles as well as outside of the heart. But the AV valves, they have lots of different names. Again, I prefer atrioventricular, sorry, because that's just so descriptive. It's very easy to remember as well. It makes sure blood flows one way from atria to ventricle and not back the other way. You have a right AV valve, also called the tricuspid, and the left is the bicuspid or mitral. This one has two names. That's fun. Remember, just try before you buy. So if you like the tricuspid and bicuspid, you try on the right and then buy on the left. So try before you buy. And then those cusps, that's describing the shape of the valve. The tips of the valves are anchored to the heart wall itself with these chordae tendineae, so tendinous cords, and specifically we call them the chordae tendineae. So you can see the tricuspid has three cusps, three little lips, whereas the bicuspid has two, and those are attached to tendons to the heart muscle. Specifically to the papillary muscles, inner wall of the heart, and this is what contracts during ventricular contraction to make sure blood doesn't flow back up through the AV valves. So blood is supposed to go from atria to ventricles. So that the shape of the cusps allows that to happen. Here you go. Allows that to happen very naturally from the atria to the ventricles. But then as the ventricles contract, they're going to be pushing blood. We want them to go out through either the pulmonary or systemic circuits. And so as blood is being pumped, it's going to force these valves closed. But then that pressure could be so much that if you didn't have these anchoring cords and muscles, the valves would prolapse, where then the pressure of the heart and the ventricles would get so much that it would pump blood back up to the atria. So by having the tips of the valve anchored to the heart wall by these papillary muscles and the tendinous cords, that makes sure that when pressure increases in the ventricles, it can only go out through the pulmonary trunk or the aorta and not up into the atria. Okay, that's enough for that. So other things, you have superior and inferior vena cava. So vena cava, these are your veins, the large ones. The superior vena cava is bringing blood from the from the head and the right arm. The inferior vena cava is bringing blood from the entire rest of the body. So they both kind of empty into veins. They get larger and larger until they connect back to those. the coronary sinus, and all of these things are going into the right atrium, these first two at least. The coronary sinus, this is referring to a large vein on the back of the heart that drains blood from the coronary circulation, so the myocardium, the blood vessels supplying fresh blood to the heart itself. The coronary sinus also empties into the right atrium. Just understand that the coronary sinus refers both to the large vein and the hole in the heart. A sinus could be a hole or a passage, and so in this case, it's representing both the vessel and the opening in the heart wall. Because if you think the spirit and inferior vena cava, they also come through holes technically, but they're connected to these large vessels. This is also connected to a vessel, but it's the coronary sinus is the name of the vessel and the hole. The right ventricle, a little thinner wall than the left ventricle because it's only pumping it to the lungs versus the entire body. And each side has a semilunar valve. So there's an atrioventricular valve and a semilunar valve. The pulmonary semilunar valve is between the right ventricle and the pulmonary trunk, that artery that's taking deoxygenated blood from the heart to the lungs. So I'm going to say that again. Between the right ventricle and the pulmonary trunk, the vein that carries deoxygenated blood from the heart to the lungs. I said it wrong. The right ventricle and the pulmonary trunk, which is an artery that takes blood away from the heart to the lungs. That's deoxygenated blood. That's why it's blue. So it's a blue vessel, so it's deoxygenated. It's taking blood away, so it's the pulmonary arteries. That goes into the lungs, gets recycled, right? And then it comes back on the pulmonary veins, which are now red because they are oxygenated, and veins because they're returning. So we don't call something an artery or a vein based on whether it has oxygen or not. Arteries are going away, veins are coming back. Good. Glad we got that settled. Okay, so let's get back into here. We're talking about the semilunar valve. So these are controlling the flow of blood from within the heart to the outside. AV valves are controlling movement of blood within the heart itself. Right ventricle, pulmonary semilunar valve, left ventricle, aortic semilunar valve. So they're named after the vessels that they're being pumped out into. And there's this picture that I've been kind of going back and forth to. You should definitely understand these images, how blood is moving through the heart, and be able to name how all of these structures, their oxygenated, deoxygenated status, and where they're going, where they're flowing, what role they play in the flow of blood around the heart. And there's just a nice little table for you if you'd like to see it written out. Okay, so blood flow proceeds in a continuous circle. Your blood, heart starts beating around, 8 weeks, and then it continues until you die. And so it's moving through these two circuits, the pulmonary circuit and the systemic circuit. Pulmonary, heart and lungs, systemic, heart and body tissues. This is all for exchanging nutrients and gases and wastes. So let's kind of walk through the step-by-step flow of blood through the heart. Again, I'm going to do this kind of quickly because we did go over this in lab, and this is something that you really need to just study rotely. It's not hard, and I think reading it out might make it a little bit more tricky than versus watching a video. So oxygen-poor blood starts in the right atrium. That's always a good place to start when you're learning the cardiac cycle or the flow of blood cycle. It's going into the right atrium through the vena cava or the coronary sinus. That's how the blood is all returning to the right atrium. Those are the three vessels that return blood to the heart. So all blood in the body is going to be filtered through, or diverted rather, through these three vessels. The right atrium contracts, so it's going to contract a little bit, forces blood through the tricuspid valve, that right AV valve, into the right ventricle. That's then going to pump it through the pulmonary semilunar valve, into the pulmonary trunk and arteries, the pulmonary arteries. Those carry that blood, that deoxygenated blood, to the lungs, where it goes into the alveolar capillaries. The alveoli, we'll learn in the respiratory lecture, are the actual places for gas exchange. They're these structures in the lungs for gas exchange. That's where carbon dioxide gets offloaded and picks up oxygen. The blood picks up oxygen. Now this oxygen-rich blood starts to come back into the left atrium through the pulmonary veins. So veins returning, volvaire return, into the left atrium. Left atrium pumps it through its mitral bicuspid left AV valve, however you want to call it, into the left ventricle. That's the really thick heart muscle-walled chamber, and then as that left ventricle contracts, it forces blood through the aortic semilunar valve into the aorta, and then through all of its tributaries to the rest of the body. Here's that kind of diagram again. You should be familiar with this. Same thing if you like to see it written out versus in an image shape. Okay, blood supply to the heart. We're just going to look at some kind of heart stuff real quick. Side story, I had an uncle a few years ago who had, what was it, a septuplet bypass? So you might have heard of people having bypass surgeries, and you know it's a big deal. You know it's a big thing, but maybe you don't quite know what it is. And it's one of those things, like, a lot of people probably don't know what's actually happening. You just are trained to know that when you hear someone having cardiac bypass, that sounds real dangerous. That sounds very serious. And it is. It is very serious. So this is what's actually happening. A lot of times when people talk about having a bypass, what you have to remember is that as the heart is pumping blood to the rest of the body, there's also that the heart is an organ itself. It requires oxygen and nutrients just like the rest of your body does. And so it has its own blood supply, its own set of arteries and veins, carrying blood to its heart wall and then draining it back into that coronary sinus. So you have right and left coronary arteries. Anytime you hear about coronary arteries, these are referring to the ones that are specifically feeding the heart, feeding and draining the heart. They're the first branches off the aorta. They carry oxygen-rich blood right to the heart. Blood flow increases while the ventricles are relaxed because they're not being compressed. So blood flow increasing during the relaxation of the ventricles as they kind of soften the vessels, or because these are moving through the heart muscle, right? So once the heart muscle contracts, you have this kind of stopping of the blood flow, and then as the heart muscles relax, blood flow can kind of release through. So it's very much a pulsing kind of blood flow that's moving through the heart. So constant pumping action of the heart, the cardiac muscle cells require a continuous supply of oxygen. So where like some muscles, you don't really use them all the time. You might not be using your glutes if you're sitting right now. They're not really needing that much oxygen, but your heart is constantly pumping, so it needs a constant supply of oxygen. And so these vessels need to always be able to supply the heart with blood. They feed, those coronary arteries feed lots of capillaries moving into the myocardium. And a lot of these arteries rather, have these connections called anastomosis. And an anastomosis is essentially, because these blood vessels are so small and they can be put under such intense pressure, it's important for there to always be a route around them. About me, I live kind of in the I live in Raleigh proper, right in the city proper. I used to live out in the country. I used to live in 20 minutes outside of the nearest McDonald's from my place, and so I lived kind of in very much. There was one road to get to my house, and when that road got flooded with a hurricane, well, that's too bad. We had to clear that road out, and that was our only path. So we had to make sure we always had a way to clear that road. Well, now I live in Raleigh, and I have 7 different ways that I can get home. So if a road is blocked, I can go around this way, I can go around that way, I can take that route. So there's always a way for me to be able to move around to still get to my home. That's what an anastomosis does for the heart. It allows the heart to make sure, because it needs this constant supply of blood, it allows vessels to be able to connect into different ways without having to really restructure the entire blood flow around the heart, while still being able to supply the tissues that need it without it being this disruption of blood flow totally. And so these are alternate pathways for blood in case something becomes blocked. Cardiac veins then drain that from the heart muscle, carrying it to the coronary sinus. So why was I talking about bypasses? This is what it is. An anastomosis is essentially a bypass. And it gets to the point sometimes where you have a large enough artery that gets blocked, or it gets so far blocked that it can't create an anastomosis, and that's when you have to come in and put in a coronary bypass. You are bypassing certain coronary arteries. They're going in and putting in artificial anastomosis, or shunts, to be able to divert blood flow from one to the other around that blocked space, and still be able to supply the myocardium of the heart with blood vessels. So when you hear about an octuple bypass or a quadruple bypass, that means they're quadruple. It's 4 coronary arteries that they're having to bypass around. Octuplet, that's 8. So that's what that is. Okay, so heart action. So let's talk about the actual contracting of the heart. The heart chambers have to function in a coordinated manner so that blood can continue to flow one way through the heart and constantly through the body. So you may have heard of these words before, systole and diastole. When you go and get your blood pressure taken, they have the top and the bottom numbers, right? And so the top number is your systole, the bottom number is your diastole. Systole is the contraction of the heart chamber. Typically, it's the ventricles that we're measuring with both of these. And the diastole is the relaxation of the heart chamber. So your heart is always has pressure. There's always blood in your heart, so it's always got this kind of latent state of pressure, nascent state of pressure on the inside of it. So while the heart is in its relaxed mode, the ventricles are relaxed. It's the diastolic pressure, which is why it tends to be the lower number, and the systole tends to be the higher number. The cardiac cycle is these events that we're coming up onto. So the cardiac cycle is what forces blood through the heart. So you learn the flow of blood through the heart. And if you don't know the flow of blood through the heart yet, you need to go back and study that. You do not need to continue on. All of this stuff is going to build because it's going to start getting pretty intense here soon. Just a heads up. So the first the atria contract. That's atrial systole, while the ventricles relax, the ventricular diastole. These things can't happen at the same time. If the atria are going to be pumping their blood into the ventricles have to be in a relaxed state to be able to receive that blood. Then the ventricles are going to contract. The ventricular systole, both of them contract at the same time. Even though you have that septum dividing left and right sides, the left and right ventricles contract at the same time. The left and right atria contract at the same time. But while the ventricles are contracting, that ventricular systole, the atria are relaxing, the diastole. And why is that's important is because during diastole, this is when something is filling up. So first up here, when the atria are contracting, right, they're full of blood, so they're pumping it into these nice relaxed ventricles. Well, while the ventricles are pumping their blood out, the atria can be relaxing, refilling up with blood, so they're ready to start this back over again as soon as the ventricles have finished contracting and are back into that relaxed state, with a brief moment in between. Okay, so cardiac muscle cells then work like skeletal muscle cells. There are a couple of differences. They have that striated appearance, but they are involuntary. You can't decide, you can't control when your heart is going to contract. They have these branching networks, though. And these kind of branches, on them, we always describe like as a Y-shaped branch, it's like a fork in the road. These are all connected by what we call intercalated discs. And intercalated discs are these structures within cardiac muscle cells that allow them to function together. Because you're having your atria contracting at the same time and the ventricles contracting at the same time, and then obviously relaxing alternately, that all has to be timed very precisely and very carefully, and it needs to be nice and even and regular. And so the heart muscle cells achieve this by doing what we call functional syncytium. And this is one of my favorite words, syncytium. There's so many great ways to know about something being syncytially, right? This is when something we say is in syncytium, it means that you have multiple parts acting as one unit. So in this case, you have all of these, heart muscle cells that are getting individual contractions or individual signals and are contracting individually, but they are doing it all at the same time because they all have these intercalated discs connecting them and making sure that when the first atrial muscle cell gets a signal, all of them are going to get that same signal at the same time. Same thing with the ventricles, the ventricular syncytium. And so these are making sure that both chambers in the atria and both chambers in the ventricles are contracting and relaxing at the same time. The syncytia, so the atrial and ventricular syncytiums, are separated by the fibrous skeleton of the heart, if we remember that structure. Okay, so let's get into the conduction system. So we kind of talked about the heart muscle cells, we talked about the heart walls, we talked about the flow of blood through the heart. So now we need to talk about how that actually is initiated, and what are the electrical signals for it, because the heart muscles, while they're involuntary, there is still a signal that's being sent from your body to initiate that rhythm. Specialized cardiac muscle conducts impulses throughout the myocardium. The cells don't contract, but they initiate and distribute impulses in the heart, coordinating the cardiac cycle. So these are special muscle cells that are embedded within the myocardium that are making sure that the contractile muscle cells are doing it at the right pacing and the right time. They're working that syncytially. So we call it the cardiac conduction system. And you may have heard of some of these structures, like the SA node. This is your pacemaker. You might have heard of people having external pacemakers, artificial pacemakers. This is what sets the rhythm of beating of the heart. It keeps things flowing, keeps things moving. A self-exciting mass of specialized cardiac muscle located in the posterior right atrium, generating the impulse for the heartbeat. Okay, so it's starting up in the right atrium. That's why it's good to learn blood flow from the right atrium. So the SA node starts in the right atrium, and that's where it sends the signal from the atrium, from the SA node. It then starts to travel down through the heart. I'm going to skip ahead just a little bit. So we're right here. I'm sorry, right up here. We're up here at the SA node. So it starts to send its signal through the atrium because they're functioning syncytially. So the SA node depolarizes and sends signal to the entire atrium. Then that signal starts to gather here at the AV node because, again, these electrical cells aren't evenly distributed through the muscles, through the heart muscle. They're in certain spots. So the SA node, all that signal kind of sends to these cells, but then it sends a signal specifically to this AV node, which then travels to this AV bundle. And this is important. This helps it to slow down, and this slowing it down allows the atria to fully contract. They get their signal, they fully contract, they pump blood into the ventricles, and then by the time the ventricles are full, this signal from the AV bundle is then moving down these bundle branches. And then the bundle branches, it moves into the heart wall to these Purkinje fibers that we talked about earlier. And then that sends these contraction signals all to the heart myocardium to contract and pump blood out of the ventricles. So keep this in mind as I go through these kind of slides about the conduction system, is that it's starting the SA node, travels through the atrium, activates the AV node, the AV bundle, the bundle branches, the Purkinje fibers. And again, this is all, these extra little steps are to make sure that, one, the heart muscle contracts evenly, and two, that the ventricles are full of blood before they actually get the signal to contract. So starting at that SA node with the pacemaker, from the SA node, we go through that atrial syncytium to the junctional fibers to the AV node. So that's our next big step, is the SA node to the AV node. Then it passes it into the AV bundle. And again, like I said, it's delaying the signal so the atria finish contracting, the ventricles can be fully filled. Then we get into the AV bundle. Sometimes you'll hear it called the bundle of His, but we do really use the AV bundle more and more. You don't hear this as much. From the AV bundle, it travels through the bundle branches and then into the Purkinje fibers. And this is what is directly actually spreading the impulse to the ventricular wall and papillary muscles. Purkinje fibers stimulates all of those muscles to contract and this upward motion, so that way it kind of starts from the apex and moves up. So it's very, I know I talked about them all operating as one, but it does kind of take this coordination, this intimate coordination, to be able to make sure that blood is being pushed that way. This sends blood from the ventricles into the aorta and the pulmonary trunk. So it's going into both systems at the same time, so the pulmonary circuit and the systemic circuit. There's that picture we just went through. And then here it is kind of written out. And there is a video on Connect, and I strongly recommend watching these videos because it's going to connect both the conduction cycle, the electrical cycle, with the flow of blood through the heart, and it gives you a video of it, so it's pretty helpful to watch that. Okay, so now let's start to look at reading those electrical signals. So that can tell us a lot about the heart activity. So that's called an electrocardiogram, ECG. If you ever see it written as EKG, it is the same thing. They're both the same thing, ECG, EKG. Truly, it's just that one is Greek and one is Latin. But it's the same thing. So it's electrocardiograms for both of them. EEG is an electroencephalogram. That's when they attach it to your head and read your brain waves. That is very different. ECG, EKG, same thing. You'll most always hear ECG now. So this is recording of the electrical changes that occur during a cardiac cycle. So you've seen them before as these graphs. So let's talk about each little point, this PQRST waves. So each one of those waves, each one of those spikes or dips is reflecting the electrical events within the heart that we just went through, the SA node and AV node, that conduction cycle. When we talk about the electrical events of the heart, we're talking about changing the resting membrane potential, the charge within those cells. So if you remember that when something has a charge to it, it's polarized. Think about like a polarizing topic, something you might bring up on Thanksgiving, right? A polarizing topic is a charged topic. So when cells are polarized, that means that they have a charge to them. And typically it's a negative charge. Inside of cells tend to be more negative relative to their outside. And so, negative 70, negative 90 millivolts is very normal. If literally they take electrodes and they can attach them to cells and read the electrical current within those cells. And so, that's a polarized cell, is one that has that negative charge. Depolarized, so you're unpolarizing it, means that it's moving closer towards zero. And some of them even go up to above, positive 30. And so that is a depolarizing process. And then there is repolarizing, where it goes back down to its negative charged potential. So just keeping those concepts in mind as we go through this. And if you need to, pause here and go and review that from the nervous system chapter. Okay, so that recording that we're looking at, this ECG, it has different waves, different components. And we can use that to actually start to diagnose things. We're not going to really get into this class, but that's what doctors will do when they read EKGs. Sorry, I said EKG, ECGs. They'll read the difference in the spikes, the height of them, the spacing between them, and that can help determine what heart issues they're seeing. So the first wave is the P wave. That is the depolarization of the atria. So as the atria are depolarizing, and go ahead and get this stuck in your brain, depolarize equals contraction. Depolarize equals contraction equals systole. I'm going to say it again. Depolarization equals contraction equals systole. They're all talking about different aspects of the heart cycle, and hopefully you can understand that. And of the heart function. So depolarization is electrical. Contraction is referring to that physical muscle, heart muscle contraction. And then systole is talking about the action of increasing pressure. So they're all slightly different aspects of heart physiology, but they mean the same thing. And it will help you, because when we get into a couple of slides, which I'm going to, of course, jump into real fast here, when we get into this slide, this is like the master slide, the primary slide for this chapter, because it's going to talk about pressure, volume changes, electrical changes, heart sounds, as well as the cardiac cycle up here, distally, diastole, and systole. And so this is going to be the chart where it's like, all right, if you can understand this and how all these things relate, you're doing real good with this chapter. So that's just why I kind of go ahead and throw that in here. Depolarization equals contraction. So in the P wave corresponds to depolarization of the atria, that leads to the contraction. Next is the QRS. That's depolarization of the ventricles. So the ventricles contracting. And then repolarization of the atria actually happens here. So the atria relaxing happens while the ventricles are contracting, but we'll talk about how you don't really see that. Mostly what you're seeing when you look at the QRS complex is just the large change in the electrical strength of the ventricles. And the last one is the T wave, and this is the ventricles relaxing due to ventricular repolarization. So here's where you got this. You have your P wave, so this is when your atria are contracting. So as the atria, so let me clarify my depolarization equals contraction statement. The electrical signal has to happen first. So right here, at the peak of the P wave, that's when the cells of the atria, the muscle cells, are starting to really get that signal, but they don't actually contract until right here in this flat portion. This is where the atria are actually contracting. So they get the electrical signal to contract, and then because electrical signals, we're talking about these milliseconds, it starts to go ahead and lose that electrical spike, but right around there, right into this flat part. This is the primary site of atria contracting. And then you have this boom spike of the ventricles getting depolarized. And because the ventricles have to pump so much harder, they have a much higher change in their resting membrane potential. And so you have this huge shift in the electrical signal coming from the ventricles. And then it's in this space right here that the ventricles start to contract. Actually, it's like right at the peak of the R to right about here is where the ventricles are contracting. It's also right within this kind of QR space that the atria are relaxing. So because, in the T wave, this is the ventricles relaxing. So the ventricles get the electrical signal to depolarize, they start to contract through here to here, and then as they get this signal to repolarize, they're going to stop contracting, again, right about the middle of the T, and then boom, this starts over again. So this line leads right over to here again, the atria contracting. But like I said, the atria are going to be relaxing right around this QR time is when they relax. So you just don't see the bump like you do for the ventricles because you have this ventricular depolarization. Everything okay? Everything for that one. This one is one that's abnormal. So this is prolonged QRS due to the AV bundle damage. So you have this kind of P wave here and this longer space for the QRS complex because it's being delayed too long. So that's just an example of how you could read those to determine maybe where a issue is happening in the heart cycle. There are also heart sounds. Your doctor, when you go to the doctor, they listen to your heart, they listen to your lungs, and those sounds can help them in diagnosing certain things. Heart sounds are lub-dub sounds. The first heart sound, that's ventricular systole, so that's the AV valves closing as the ventricles contract. They push blood out through the semilunar valves, and the AV valves close up, so that's that lub. And the second sound, the dub sound, this is the ventricular diastole. So that's the aortic and pulmonary valves closing back up because they're pretty high pressure too. So they have that dub sound. So lup, dub, lup, dub. Any abnormal sound, we call it a murmur, usually due to valve damage of some kind. Okay, so there's also pressure and volume changes during a cardiac cycle. As the heart is contracting and relaxing, that's changing those pressures within the heart. So during the cardiac cycle, pressures change within the chambers, it rises and falls, as well as those heart valves open and close. It helps them to maintain their proper movement so that blood doesn't flow abnormally. Early ventricular diastole pressure in the atria is greater than that of the ventricles, and that forces the AV valves open, allowing the ventricles to fill. So yes, the atria do have a slight contraction to them, but it's also just the fact that the ventricles are fully relaxed, so they're really relaxed. The atria don't have to be, because they're already filled with blood, it's just kind of primed to be able to push that blood, forcing the AV valves open. So 70% of blood flows passively from the atria to the ventricles before the atria even start to contract. So that's just due to the difference in the pressures. After the atria depolarize, the atria pressure rises even more, and that pushes the remaining 30% into the ventricles. During ventricular diastole, that pressure increases sharply. Sorry, hold on one second. I'm trying to make sure if this is written correctly or if this is what it wants to say or if this is a typo. During ventricular diastole, pressure inside them increases sharply, causing AV valves to close and the aortic and pulmonary valves to open. So this probably is not supposed to say during ventricular diastole because that would be them relaxing. What it might be talking about is that this is what I'm guessing here, is that so it's saying during ventricular diastole, this is referring to the end of the diastolic cycle while the ventricles are almost filled with blood. So they're still relaxing, but because they're filling with blood, the pressure inside them is starting to increase. So I'm going to teach this as if we are talking about right at the end of this atrial depolarization. We just got that last drop of blood from the atria into the ventricles, but the ventricles haven't started to contract yet. They're still in this relaxed state, but because they're filled with blood, the pressure inside is increased enough to close those AV valves, so it seals off that passage to the atrium, and then it starts to push open these aortic and pulmonary valves. Then you have the ventricular systole, where the muscle cells actually start to contract. Those papillary muscles contract, holding the AV valves closed, and then blood gets pushed out of the ventricles into these semilunar valves, the aortic and pulmonary semilunar valves. And as that happens, the pressure starts to drop because the ventricles are pushing this blood out. So it's kind of like, I've never been a fan of the Dr. Pimple Popper videos myself. But we have all had that sensation of popping a pimple, and you can feel how, even though it takes, you increase the pressure, once it actually pops, then the pressure decreases, even though you're still pushing, because now it's no longer full. So that's what's happening, is that blood gets pushed out, but the pressure is dropping because that blood is being pushed out, and so then the ventricles start to relax. Once ventricular pressure is lower than blood pressure, so blood pressure being in the vessels, the aortic and pulmonary and pulmonary trunk, not truck. The aorta and pulmonary trunk, those semilunar valves start to close. So again, the semilunar valves look just like the AV valves, except they're making sure blood doesn't flow from the aorta and pulmonary trunk back into the ventricles, back into the heart. Once the ventricular pressure is again lower than atrial pressure, the process happens all over again. The AV valves are going to open because during this time of the ventricles emptying, the atria are filling, and so this. As soon as that pressure changes, shifts again, the AV valves open that passive flow. So here we are at this chart once more. So again, you should be able to go through this. I'm going to try to zoom in a little bit. You should be able to go through this and follow all of these different changes. You're looking here at the aortic pressure, ventricular pressure, and atrial pressure in the brown line. And so you can see this big one, the one that has the biggest pressure change, that's going to be the ventricles, because they're having the biggest shift in, they have the strongest heart muscles, so they're going to have that biggest change in pressure. Right, and then it starts to relax here, and then right around here, the pressure starts to increase. And what happens? Why is the pressure increasing in the ventricles? Oh, well, the atria are contracting, and they're filling the ventricles up with blood. Now, if I go down here and I follow this little increase in ventricular pressure with this increase in atrial pressure, what happens to my volume? Oh, my ventricular volume is starting to increase right here because it's starting to have this atrial contraction, pushes blood into the ventricles. And then what's happening here? This ventricular blood volume is really starting to drop. Oh, the pressure is increasing in the ventricles. I'm in ventricular systole. And then what do you know? I'm right after my QRS, my ventricular depolarization. So you can just go on and on. You can follow all of these lines through each of these different graphs, and you should be able to understand that. That's a good way to kind of check yourself to see if you understand the system so far. And do it like I did, kind of making it a story and kind of talking about the specifics that are happening here, not just reading it and saying, oh, well, look at the volume shift from 130 milliliters to 160. No. And this is just another way to be able to kind of track that cycle. All of those cycles. All right. Take a breath. This is an intense chapter. Let's kind of acknowledge that. So if you need to take a break, pause this video, come back and finish it when you're ready. Maybe just stretch and get some water because we still have some pretty in-depth stuff to get into. So heart rate and regulation of the cardiac cycle. This is all dependent upon the activity level of somebody at that moment. The more active you're being, the more blood flow you're going to need to have. So average adult resting heart rate is 70 to 75 beats per minute. Normal, though, is 60 to 100. So if it's within that range, it's still considered normal, and that's why it's nice to kind of make sure, especially as you're younger, you're checking your blood pressure, you're getting all those things done, because then you can understand what is normal for you. What is your normal heart rate and blood pressure? Anything over 100 beats per minute, though, is considered tachycardia, so an increased heart rate. Anything less than 60 beats per minute, bradycardia, a slower heart rate. This can be, especially with the bradycardia, because if your heart is able to contract stronger and more forcefully, it doesn't have to beat as fast. And so that's where you kind of get this large range of beats per minute, is because, well, if your heart muscle is maybe a little bit thicker, a little bit stronger, it can contract with more force and not have to beat as much. As we said, it adjusts based on the current needs of the body. Heart rate changes in response to factors affecting that SA node, so the sinoatrial node, that pacemaker. Sympathetic or parasympathetic stimulation can start to shift it. Sympathetic impulse increases the speed and strength of heart contractions, so your fight or flight is going to cause your heart to pump more. Heart rate decreased by parasympathetic impulses, slowing you down, rest, and digest. The cardiac center in the medulla oblongata, part of the brainstem, maintains this sympathetic-parasympathetic balance. There is the cardiac center, the vasomotor center in the medulla oblongata. Sorry, hold on. And both of these help to maintain proper blood flow, blood pressure, cardiac output, all of that. The cardiac center is responding to what we call baroreceptor reflexes. A receptor, so this is a nervous system thing, a receptor, and baro refers to pressure, like a barometer. So a baroreceptor reflex detect changes in blood pressure in your vessels. Impulses from the cerebrum or hypothalamus can also influence it. So emotions, if you are very scared or you're anxious about something, that can increase your heart rate. Changes shifting from the hypothalamus, thyroid hormone, any other types of growth hormone, regulatory hormones can start to shift your heart rate. Thinking like epinephrine release, all that kind of stuff. Increased body temperature increases heart rate as well. Certain ion concentrations can affect heart rate. Hyperkalemia, so excess amounts of potassium, decreases heart rate, can lead to life-threatening arrhythmias. Potassium is very tightly regulated in the body. Same thing with hypercalcemia, that increases your heart action, whereas hypocalcemia, to not enough calcium, decreases heart action, depresses heart action. There's just some looks at how the heart rate is influenced, if you like to look at those pathways between baroreceptors, the carotid sinus, carotid baroreceptors, aortic baroreceptors, detecting the pressure changes, and then relaying those to the medulla oblongata, which then sends signals to the SA node to adjust its firing patterns. All right, blood vessels. So we're talking about blood pressure, so we start to talk about blood vessels. Blood vessels formed a closed circuit, so we have a closed circulatory system as opposed to things like insects that have open circulatory systems. This carries blood away from the heart to the cells and back again, traveling on arteries, arterioles, capillaries, venules, and veins. Arterioles are small arteries, venules are small veins. Arteries, they're strong elastic vessels adapted for carrying high pressure blood. Think about the aorta. It is that vessel that is directly attached to the heart. It's under high amounts of pressure, so it has to be able to expand to be able to adapt to that shifting pressure. Arteries transport blood away from the heart, and as they become smaller, they divide and give rise to arterioles. There's 3 layers to an artery, and to some of the larger arterioles as well. The tunica interna, media, and externa. The interna is the inside of the blood vessel. It's that epithelial tissue, has a smooth surface to prevent clots, secretes biochemicals to prevent platelet aggregation, and substances to regulate blood flow. So this is that actual inner lining of the blood vessel that's attached to the lumen or touching the lumen. The media is the middle layer. It has smooth muscle. And the externa is the outermost connective tissue, kind of holding it in place, making sure that it's attached to the right spaces, moving along the correct track. Sympathetic impulses cause that smooth muscle walls to contract. So through the vasomotor fibers, it causes vasoconstriction of the arteries. When those signals are inhibited, you have vasodilation, so a relaxing opening of the blood vessels. Vasoconstriction increases blood pressure. That's going to help make sure more blood is being pumped and moved to the body during fight or flight times, whereas vasodilation decreases blood pressure. That can be a slightly confusing concept. Think about a water hose. If you have a water hose and you're trying to increase you want to spray a little bit more, you might put your thumb on the end of the water hose to have an increase in pressure and faster blood flow. You might have a little less water coming out, but it's coming out at a higher speed, and so it's transporting faster. So it's transporting oxygen faster. Whereas if you were to move your thumb, and that water might just kind of trickle out. So it may be flowing more water, but it's not actually flowing fast enough. So vasodilation drops blood pressure and causes slower blood flow rate, whereas vasoconstriction increases blood pressure, increases blood flow rate. And sympathetic control regulates blood flow and blood pressure. Larger arteries have walls, some arterioles have walls like arteries, as I was saying, but as they get thinner, as those arterioles divide and they get more thinner and get to capillaries, they may only have the endothelium, a few muscle cells, and some small connective tissue layers. Here you can see the artery versus a vein. So they both have kind of this tunica externa, that connective tissue layer anchoring them in space. The tunica media, you can see how thick it is on the artery versus on the vein. And then veins also have on their endothelium, the tunica interna, they have these valves where arteries do not. Yeah. So here's an arteriole. So this is one of the divisions of the arteries. You can see these smooth muscle cells that are wrapped around it to help for vasoconstriction and dilation. And this little branch right here is a capillary. So this is leading off into a capillary or maybe even a capillary bed, who knows. But it has this structure called a precapillary sphincter. And so these can be constricted or dilated as needed to divert blood flow. And we'll look at that in a moment. So capillaries then. So hopefully you can see and understand how this chapter is being structured. Where we started at the heart, we talked about all the ways blood flows through the heart, how it happens, the electrical signals for it, and now we're following that blood out of the heart into the body, and we're talking about how blood actually gets into the tissues. So from the heart into arteries, from arteries, arterioles into capillaries, and now we're in our capillaries and capillary beds. And then we'll move into veins back to the heart. So capillaries, blood vessels with the smallest diameter, connecting small arteries and arterioles and venules together. They only have a layer of endothelium, so usually it's just one single cell layer thick that allows substances to be exchanged, gases and waste and nutrients, hormones, proteins, blood cells, white blood cells, things like that, fluids, water, all that can be exchanged. Capillary permeability varies from one to the next due to the size of openings between the cells. So in places like the kidneys, the liver, intestines, endocrine glands, smaller muscles, and endocrine glands, they tend to be larger because these are places of filtration. Your kidneys, your liver, your intestines, they are constantly filtering your blood, making things, sending it back out, absorbing nutrients, and so they tend to have slightly larger openings in their capillary bed. Same thing with endocrine glands, they're releasing hormones, so they have larger openings to allow things to exit or enter as needed. Muscle tissues don't really do that. They don't do any blood filtration, and they may produce some waste, but they're not producing large hormones or proteins. So very small openings. They really only need to have that exchange of the needed nutrients, glucose, oxygen, water, and the removal of waste. Here you can see a sample capillary cell. Tissue fluid moving in and out through these openings, these pores or slits, as you want to call them, and these endothelial cells kind of forming the structure of capillaries. So capillary density varies from one body part to the next. Areas with a lot of metabolic activity, things like your leg muscles, have more capillaries, while others that aren't as metabolically active may not have quite as many capillary beds. What's a good example would be something like maybe the muscles in your eye, right? They're pretty small. They do small movements. And a lot of times you're turning your head. If you're going to look at something over to the side, you're probably eventually going to turn your head so that your eyes are pointing straight ahead. And so they tend to not have to work a whole lot unless you're just really busy rolling your eyes. So density is related to metabolic activity. More vascularized, the more active something is going to be. Some are highly branching networks, these big intricate capillary beds, while some can just connect an arteriole to a venule, just one, kind of shifting down to a capillary and then back up to a vein. I mentioned those precapillary sphincters before. They regulate blood flow entering a capillary bed. If blood flow is needed elsewhere, those capillary beds, for the less important areas, can receive less blood flow just by constricting and contracting that precapillary sphincter. During exercise, for example, capillary beds and skeletal muscles open up through that vasodilation and relaxation of the sphincters, while the blood vessels to your intestines and to your your intestines specifically can start to constrict and divert all that blood flow that would be in your intestines to your skeletal muscles, and then vice versa. When you're relaxing, when you're not working out or doing anything super active movement wise, your blood will shift from your muscles back to your intestines. And during sleep, while we're sleeping, up to about 80% of your blood can be diverted to your intestines to allow for nutrient absorption and exchange. This is a capillary network. You can see arterioles moving into the capillaries and how they connect into veins. So exchanges in capillaries. It's a constant exchange of respiratory gases, nutrients, metabolic wastes. between capillaries and tissue fluid near body cells through diffusion, filtration, and osmosis. So take a moment and read that again if you need to make sure that you understand everything that's being shifted and the processes through which it's being changed, diffusion, filtration, osmosis, and the fact that it's not going directly to the body cells. It's between the fluids, the fluid within the blood and the interstitial fluid outside of the blood vessel, outside of the cell. That's where things are being shifted, and then they filter, either drift into the cells or into the capillary beds. Blood that enters capillaries has high concentrations of oxygen and nutrients. So entering meaning coming from arteries into capillary beds, not from interstitial fluid. But blood that's coming through capillaries has high oxygen, high nutrients, and that will diffuse through its concentration gradient from the capillaries into the tissues. Let's say you're dehydrated and you're drinking some water to help yourself rehydrate. You're going to have a higher concentration of water in your capillaries, and so that will, through osmosis, right, a type of diffusion, through osmosis, it will leak out into the interstitial fluid and into your cells. And as long as you're also making sure you replace electrolytes as you're replacing fluid, then it will stay balanced. Plasma proteins, they tend to remain in the blood due to their larger size. So remember those proteins that are hanging out in blood vessels. I'm sorry, or in the blood, those albumins and globulins, things like that. They'll tend to stay there unless they're getting certain signals that will allow them to move through capillary beds. Carbon dioxide, metabolic wastes, they go from the tissues, from the interstitial fluid, into the capillaries because there's going to be a lot less carbon dioxide and metabolic waste in the capillary beds. So they will naturally diffuse into those spaces. Again, passive actions depending on concentration gradients. You also have hydrostatic pressure from the pumping action of the heart. Hydrostatic pressure is your blood pressure. You can kind of keep those two same things in mind. Hydrostatic pressure is your blood pressure. There might be some subtle differences in there, but for the most part, we can use those interchangeably. This allows for filtration of substances through the walls of the capillaries. So filtration is an action under the it requires a pressure gradient for it to happen versus a concentration gradient, which is a passive action. Filtration, pressure gradient, active. Blood pressure moves blood through the lumen of arteries and arterioles. So that blood pressure, that hydrostatic pressure from the heart, is what keeps blood moving through those vessels. But it does decrease with distance from the heart. So blood pressure stays greatest in the arteries, lowest in the capillary, or lower in the capillaries, and even lower in veins. On the arterial end of capillary beds, blood pressure tends to be a little higher than in the venous end, the venular end. So at the arterial end, hydrostatic pressure, your blood pressure, is driving filtration of fluids and very small molecules out of the capillary bed, but diffusion occurs all along the capillary. So again, diffusion is independent of pressure. It's going to be a concentration gradient. So that's a different force, right? Colloid osmotic pressure is generated by the presence of those plasma proteins. We said plasma proteins tend to stay in the blood. They do not filter out. They do not diffuse out. And so they stay in the blood and draw water back into capillaries. So this becomes important at the venular end where some of the fluid has diffused out. So at the arterial end, you have this hydrostatic pressure that, yes, it is pushing oxygen and other substances out, but it's pushing a lot of water out. And so as that water gets pushed out, the plasma protein concentration relatively becomes higher. And that's what colloid is referring to. Colloid is a protein-rich fluid. And so as fluid gets pushed out of the capillary beds into the interstitial fluid, your colloid osmotic pressure starts to increase because these plasma proteins are now saying, hey, there's not as much water in here. We're kind of like all having to stick together and getting clumped up. Can we get some more water in here? And so as blood moves down the venular end, it starts to draw some of that water back into itself so that way your blood vessels don't get dehydrated. Or you stay overhydrated, you have swelling in your tissues because you've just got all this fluid leaking out. So hydrostatic pressure and colloid osmotic pressure are opposing forces within your blood vessels. Hydrostatic pressure and colloid osmotic pressure are both pressures being exerted from within the blood vessel, but they do different things. Hydrostatic pressure increases filtration, colloid osmotic pressure increases reabsorption. Hydrostatic pressure increases filtration, colloid osmotic pressure increases reabsorption. So, at arterial ends of capillaries, blood pressure is higher than colloid osmotic, so filtration occurs to a greater extent, and it's the opposite at the venular end. COP is higher, so reabsorption occurs. More fluid leaves capillaries by filtration than does return by osmosis. But that excess fluid that is not picked back up is picked up through your lymphatic vessels, which we'll learn in chapter 14. And that returns it to circulation after kind of sending it through some filtration processes. Here's a depiction of that. Here's your arterial end. Here's your venular end. This is your capillary. So you can see you have this outward force of hydrostatic pressure, blood pressure pushing fluid out, inward force of the osmotic pressure pulling fluid back in. But because you're at this arterial end where you have higher blood pressure, the net pressure is going to be 11 out. Through the capillary bed, you have this inward. Now you're kind of moving through here. You've had a net loss of fluid. So now it's the COP is higher. The COP now is 24. The COP relatively is 24, and blood pressure, because you're on the venular end, is lower. So now it's only 16. So now you have this net pressure of 8 millimeters of mercury being pulled back in. And so this allows some water to be reabsorbed. Whatever isn't is restored to the lymphatic vessels. And then blood can continue to the venules. Okay, so now we're in our veins, our venous system. Venules leading from capillaries merge to form larger veins that return blood to the heart. Walls of veins have similar layers like arteries, except they have those valves to prevent backflow of blood. The lumen of the vein is larger than that of an artery because it doesn't have that thick muscle wall, so it has more of a large lumen. And blood pressure, that then means, is lower in veins than it is in arteries. You lose a lot of your blood pressure in your capillary beds, and that is not then transferred back into the veins. Veins also function as our blood reservoirs. That means that they kind of hold blood for us. So just in normal times, constriction of veins in times of blood loss can restore normal blood pressure after 25% of blood being lost to a hemorrhage. So if you've had 20, you can lose a quarter of your blood, and your veins have so much blood just stored in them, hanging out typically in your legs and your lower body, that they can cause shifting. They can shunt that blood around and restore a lot of that. So that's pretty impressive. But again, when you're not working, when you're kind of sitting and maybe listening to this lecture or laying in bed, your blood is going to collect in your veins because it's not being forced back out. So here you can see the lumen, this inside of these vessels. You have a venule up here and an arterial down here, and yeah, those are blood vessels or blood cells. You can see little neutrophils in there too. So valves and muscle contractions helping venous blood return to the heart through your skeletal and your respiratory pumps. So here's a relaxed skeletal muscle. You can see the valves are closed. Nothing's really happening. Blood's kind of hanging out here. But now the muscle contracts. It increases this pressure. And because these valves can't open downward, they can only open upward, they return blood back to the heart by compressing in these little sections. That's what veins have all along them in intervals. They have these valves, these one-way valves, that allow blood to constantly push back up towards the heart. Nice little charts for you to be able to go through the different types of vessels and their functions. All right, now let's get into some blood pressure stuff. So blood pressure is a force exerted by blood against the inner walls of vessels. It exists all throughout the cardiovascular system to much greater or lesser extents in certain areas. But typically, because of that large change, when we talk about blood pressure, we're usually referring to systemic arterial pressure. So the blood pressure in your arteries. Think about if you were to take your pulse on your wrist or in your neck. That's typically what we're talking about. Is the blood pressure, or like when they put the pressure cuff. That's a better example. When they put the pressure cuff on your elbow to take your blood pressure. I've had some put them on my wrist. The blood pressure cuffs on your wrist. You're measuring that arterial blood pressure. So arterial blood pressure rises and falls according to the pattern of the cardiac cycle. Systolic pressure is the maximum arterial pressure during ventricular contraction, whereas diastolic is that ventricular relaxation just before the next contraction. But again, it's not no contraction, or it's not no pressure, because there's still blood in these vessels. So there's still this latent pressure against the inner walls of the vessel. A sphygmomanometer, that's the blood pressure cuff. Sphygmomanometer, yes, I did say it right, is used to measure your arterial blood pressure. No greater than 120/80 at rest is considered normal. That's a number that they use. But again, it's going to vary with a lot of people. So taking your blood pressure regularly and consulting with your physician, especially if you're experiencing any secondary symptoms, is important. Blood pressure decreases as distance from the left ventricle increases. There you go. We talked about some of this already. The alternating expansion and recoil of the wall of the artery as ventricles contract and relax can be felt as the pulse. So you can take your pulse on the radial artery, carotid artery, brachial artery, femoral artery. You should know where all of these are based on their naming. So your pulse is that beating of the heart, right? The contract and relax of the ventricles. That's what you're feeling there. So this is your pressure gauge, the inflatable cuff. And then with this, you can kind of listen to and count the pulse. This is just showing how pressure decreases from the aorta all the way to the vena cava, where blood is being returned to the heart. So it's almost at this point like a passive process. All right, so arterial pressure depends on many factors: cardiac output, blood volume, peripheral resistance, and blood viscosity. We're going to get into all four of these in these next slides. Starting with cardiac output, CO. Your cardiac output is a combination of your stroke volume and your heart rate. So the heart rate, that is how fast your heart beats. Okay, your pulse rate, your heart rate, same thing. And the stroke volume is the actual amount of blood being pumped at one time. So that's how strong your heart is. So again, the stronger your heart, the lower your heart rate needs to be to meet a cardiac output. Cardiac output is the amount of blood that is required to keep your body functioning. So cardiac output can change depending on your level of activity. So as you are at rest, your cardiac output is lower, your stroke volume and heart rate can be lower. As you start to go into exercise, your stroke volume can increase to meet the needed cardiac output, or your heart rate can increase to meet the cardiac output. But something has to increase to be able to reach that. For a lot of us, especially if you maybe aren't working out as much, or you aren't, I'm thinking like an Olympic athlete, that's tend to be where I'm like, well, there's either just me that kind of works out sometimes, and then there's Olympic athletes, and there's nobody in between. So the stroke volume, and I'm not saying I'm anywhere close to an Olympic athlete. I am the couch, Anyway, the stroke volume, it's harder to get that to increase because that's the conditioning in your heart muscle. This is actually making your heart muscle stronger. It's a lot easier just to cause it to fire faster. And so as stroke volume can increase, heart rate doesn't have to increase as much. Average cardiac output is about 70 milliliters per beat, 72 beats per minute, so about 5 liters a minute or so, which that's a lot of blood. Stroke volume or heart rate increases, so does cardiac output. So those things are kind of all dependent on each other. So let's look at blood volume. So we said that your blood pressure, arterial pressure, blood pressure, cardiac output, blood volume, peripheral resistance, and blood viscosity. So blood volume is going to affect your pressure. The higher your blood volume, the higher your blood pressure. Very straightforward. These are directly related. Increased blood volume, increased blood pressure. Things like some of the volume of the plasma and formed elements. So, we talked about people using EPO in the last chapter and blood doping, how that increases your red blood cells. Well, that increases the volume of your blood. And so that can cause your blood volume and your blood pressure to increase. Your hydration status, if you are dehydrated, then your blood pressure is going to have to increase because your blood volume is decreasing. And so it's got less blood to pump, so it has to pump even more. That was a bad example. When your blood volume decreases, your blood pressure decreases. But what does happen is that it varies with age, body size, and sex. So males are going to have higher blood volume than females, except for when females are pregnant. Body size, the larger your body, the larger your required blood volume. And with age, actually, it decreases. Your blood volume tends to decrease once you reach a certain age. From childhood to adulthood, it's increasing. And then once you start to get into kind of a senior age, a more advanced age, then your blood volume will start to decrease, actually. Average adult blood volume is about 5 liters, 8% of your body weight. Blood pressure directly proportional to blood volume. So as blood volume increases, blood pressure increases. Uh, Things that can change blood volume. Oh, sorry. So let me go back to that. What I was saying before about when you're dehydrated, your blood pressure will increase. What I meant to say was that your heart rate will increase. So as your blood volume decreases, if you are not rehydrating to replenish that, your cardiac output has to correspond somehow. You still have to be able to get nutrients to your cells, and so something will increase. And in that case, it tends to be heart rate. If there's not enough blood to pump, it'll just pump what it does have even more. Factors that change blood volume also affect blood pressure. Hemorrhage or dehydration lowers blood volume and blood pressure. So whether you are bleeding out or have been working out, you've been outside doing something and you're sweating a lot, all of those things are going to start to make your blood pressure drop. Peripheral resistance. So peripheral resistance refers to the resistance of blood flow in your systemic vessels. So kind of out in the arteries and the veins of the body, how easily blood can flow through those. And the more vessels you have, that affects your peripheral resistance. So friction between blood and the walls of blood vessels. Peripheral resistance hinders blood flow, so blood pressure has to increase as peripheral resistance increases. So these are directly related. Factors that change peripheral resistance affect the blood pressure. So as PR increases, something like sympathetic stimulation, your blood pressure will go up as well because it has to overcome this PR. Because again, your body's need for that blood is still going to be consistent. So your body still has to overcome this factor to be able to get blood where it needs to go. So it does that through blood pressure. Something like vasoconstriction in arterioles can cause blood to back up into arteries. That will cause an increase in blood pressure. Plaque deposits on blood vessels that narrow the lumen can increase peripheral resistance, and that can cause an increase in blood pressure. Having excess blood vessels, so if you have a tumor or something, that's causing more blood to be lost in certain areas, more peripheral resistance, and so increase in blood pressure. And then blood viscosity. Viscosity refers to how thick blood is, or for how thick a fluid is. So the greater the blood viscosity, the greater resistance to its flow, the greater the blood pressure. So blood cells, plasma proteins, those can all increase blood viscosity. So here are those factors. Blood volume increases, heart rate increases, stroke volume, peripheral resistance, blood viscosity, all of these increases will affect positively on blood pressure. Okay, so controlling our blood pressure then. Blood pressure is determined by cardiac output and peripheral resistance. So we said cardiac output was heart rate times stroke volume. Well, blood pressure is cardiac output times peripheral resistance. So meaning that one of these is going to be shifting to maintain the proper blood pressure. Or one of these is going to be shifting irregularly and your blood pressure is going to go up. So the body maintains normal blood pressure by adjusting cardiac output and peripheral resistance. Some aspects of peripheral resistance can't be changed, but some can. Essentially vasodilation, vasoconstriction. As the body adjusts those, that can then increase or decrease blood pressure. The Frank-Starling law of the heart is the relationship between cardiac muscle cell length and the force of contraction. So what this is basically saying is that as you have more blood coming back to the heart, your heart is going to expand more and it will contract even harder. Your stroke volume will increase as the amount of blood return, the venous return increases. Whereas if you have a lower amount of blood coming back to the heart, it's not going to contract as hard. And really that comes from the alignment of the cells. With like your skeletal muscles, if you're ever doing a workout, let's say you're doing bicep curls, if you only let your arms come down, your elbows come to a 90, and then you bring you go into flexion. If you're only going down to 90, you're kind of keeping your muscles partially contracted. And as you fully extend your elbows and then contract, by fully extending the elbow, you're stretching that muscle and it's going to have a harder time to contract. That's not true with heart muscle. With heart muscle, as it gets more stretched, it actually can contract even harder. And that's to overcome this excess return of blood. This is to make sure that the heart essentially doesn't explode. So if you have a lot of blood coming back into the heart at one time, you have this increase in venous return, the heart can expand, and then it can contract equally to pump that blood back out to the body. So that way you kind of always trying to, again, maintain this, normal flow of blood. Output and peripheral resistance, partially controlled by baroreceptor reflexes. So baroreceptors sensing changes in that BP. In the aortic arch, carotid arteries, we saw they send signals to the medulla oblongata. That can initiate the cardioinhibitory or cardioacceleratory reflex. So if you have arterial blood pressure increasing, those baroreceptors are going to detect it. The medulla oblongata sends parasympathetic signals to the SA node, and that decreases cardiac output and blood pressure. So blood pressure increasing, cardioinhibitory reflex. Blood pressure decreasing, you get sympathetic signals to the heart rate, increasing CO and BP. So cardioacceleratory reflex because of decrease in blood pressure. And again, here's a nice little flow chart for you to be able to follow through with that. All right, so other things, emotional upset, exercise, raising in temperature can change the cardiac output and increase blood pressure. Peripheral resistance also controls blood pressure. So changes in arterial diameter, vasodilation or vasoconstriction can do it. Sympathetic nerve changes responding to blood pressure changes. Blood vessels with smaller diameter provide greater resistance. So as you have, again, like things like tumors, they tend to be very rich in capillary beds, have a lot of capillary beds. Capillaries mean you have small arterioles. That means small blood vessels with small diameters, more peripheral resistance. Vasodilation decreases resistance and blood pressure. Vasoconstriction increases resistance and blood pressure. Think about the water hose example again. And sympathetic stimulation causes vasoconstriction. So some of this stuff is kind of repeating itself, so hopefully we only have a few slides left. So if your eyes are starting to cross, it's okay. Come back and watch this again or watch another videos you need to, because this stuff can get a little confusing. And then you have the vasomotor center in the medulla oblongata as well. This is what's adjusting the sympathetic impulses to the smooth muscles and arterial walls. Certain chemicals can do it as well. Epinephrine and norepinephrine can do that. Some nitrogen chemicals can do that. Increased carbon dioxide, decreased oxygen, decreased pH, all cause vasodilation in tissues. And I was saying epinephrine, norepinephrine causes vasoconstriction, increasing peripheral resistance and blood pressure. And then venous blood flow. Blood pressure decreases as distance from the heart increases, so you don't have blood pressure. You can't rely on that to get blood from the veins back to the heart. So you're relying on the contractions of skeletal muscle. So moving your arms, moving your legs, walking around, that contraction relaxation pushes blood up those veins and through the valves. Respiratory movements from your diaphragm and your thoracic cage. Expanding and contracting, that decreased pressure in the vena cavas helps to pump blood back to the heart. And venoconstriction, initiated by sympathetic reflexes when venous blood pressure is really low. So that is monitored somewhat, and so you can have some constriction in the venous blood vessels. And as we've said, veins act as a blood reservoir. So venoconstriction can help to send some of that blood out of the reservoir when it's ultimately needed. All right, thank you very much.